Good afternoon, everyone. I'm from NASA Glenn Research Center in Cleveland, Ohio, and I'm working on a project that's called the Digital Astronaut Project. So the Digital Astronaut Project is within the NASA Human Research Program. And the NASA Human Research Program is designed to identify the risks to human health during spaceflight. And it's tasked with finding ways to mitigate those risks and to maintain human health and performance during spaceflight missions. So the Digital Astronaut Project provides computational modeling support to the Human Research Program. And we have a project team that's made up of civil servants, support service contractors, and academic collaborators. Our portfolio of models, it spans several different physiological systems. We have musculoskeletal system models, and then we also have muscle and bone models. We have cardiovascular and associated fluid models, and then we also have some ocular models, a heart model and kidney model, and then we have some vestibular system models. So for our musculoskeletal modeling, we have biomechanical data collection capabilities. We use biomechanical modeling software with that data. We use that information to characterize spaceflight exercise. And some of the recent projects that we've had were to look at the volume that exercise takes up and to determine if exercise was going to fit within the new Orion capsule. Also within our musculoskeletal modeling, we have physiological model of bone that predicts the decrease in bone mineral density over the course of a space flight mission. And that is used in conjunction with our bone fracture prediction models. They're used to predict the probability that an astronaut will suffer a fracture during spaceflight. And then just recently, we've added a grant to the University of Virginia, Sylvia Blunker's the PI, to study muscle atrophy and to work on a model of muscle atrophy over the course of a spaceflight mission. For our cardiovascular modeling, we have a lumped human body model that includes the cardiovascular system, cerebral spinal fluid, lymphatic fluids. And that's used in conjunction with a lumped eye model that predicts interocular pressure, and that's also used in conjunction with a finite element model of optical nerve receptors. And these models are being applied and being used to help study the visual impairment in intracranial pressure syndrome, which is the syndrome where astronauts are coming back from long-term space missions with vision impairment. So the funding, all of NASA's funding opportunities is through a website that's called NSPIRES, which I have listed here. The Human Research Program, their funding opportunities are listed as the HE HURO opportunities, human exploration research opportunities. And then we used to have a collaboration called the NSBRI, which was a academic collaboration. It was the, another arm of HRP. That has since been changed to the Translational Research Institute, which is a new collaborative effort. And I have a link to that information there as well. That's it.
Thank you, Beth. Next up is Dr. Michele Grimm from the NSF and presenting her project. And
thanks, Guy.
And just want to remind you that all these presentations are on the wiki for you to refer to all these funding initiatives, so you don't have to madly write down everything.
All right. Okay, so want to tell you a little bit about what's available with NSF. These aren't necessarily new initiatives, but want to keep everybody in mind that they exist. So the programs I'm going to be talking about are within the Engineering Directorate. And I'm hoping that I don't see any hands raised, but how many people think that NSF does not fund anything related to human health? You're going to owe me a drink at SB3C the way we're going. Okay, so obviously that's false. There are 12 different programs within the Engineering Directorate that support biomedical engineering related research. And many of these are going to support modeling approaches to investigating questions relevant to biomedical systems and human health. Funding mechanisms that we have, the interestingly named unsolicited proposals is our standard mechanism for all investigators. That is funding at about $100,000 a year for 3 years, including indirect costs. So we do not have lots of money, but it can be money that can help you get projects moving forward. The career awards are for junior investigators, so untenured but tenure track, and that's 5 years, but at the same $100,000 a year level. And then there are some special solicitations that come up every now and then to keep an eye open for. The programs I want to mention today are specifically related to multi-scale modeling. First, we have renamed the biomedical engineering program to be Engineering of Biomedical Systems. That was because everything that was BME related tried to come in through my program, and so we're trying to differentiate and make sure everything has the right home. The objectives of the program are to develop novel ideas into transformative solutions and advance both engineering and biomedical sciences. One of the key things that the EBMS program will support is development of validated models of both physiological and pathophysiological systems, tissues, and organs. And this can be in vitro or in silico. We do want to look for that validation, which we've been talking about the last 2 days. And they can either be for understanding basic principles or for more translational application. Biomechanics and mechanobiology is in a different division, so I'm within CBET. Biomechanics and mechanobiology is within CMMI, but again, they support the multi-scale modeling with a particular emphasis on mechanics. EBMS will support all aspects of engineering, and they're looking at integration across the various scale levels, and they want to see fundamental advances in the mechanical sciences. And then the previously named biotechnology and biochemical engineering is now cellular and biochemical engineering, and this is where this focus will be predominantly on molecular and cellular engineering, with a particular focus on how to translate knowledge to biomanufacturing processes. Review criteria, we have 2: intellectual merit, which maps very well to what NIH expects, but then also broader impact. And at least for the programs that I oversee, we don't allow PIs to simply point to the fact that their work will eventually improve human health to be their broader impact, because if it didn't do that, it shouldn't be in the program to begin with. So please think about how you are going to look at educational opportunities, public dissemination of knowledge, scientific literacy, something that goes along with your research, but that actually is going to have a positive impact on society. Keys to success. The first one is identifying the right program. So please reach out to program directors early and have a conversation with them about which program best fits your research, and make sure you don't just tag or reformat an NIH proposal to be an NSF proposal. All of our panelists recognize that very quickly and don't really take kindly to it. I don't care whether it's an objective or an aim, but if the broader impacts portion isn't in there, then that's definitely going to be a bad sign. And with that, I will thank you. And if anybody's interested in serving as a panelist to learn more about the way that NSF reviews things, just let me know. And I'm always interested in getting more individuals involved with the review process.
Thank you, Michele. The same is true when we see NSF formatted applications coming to NIH. We want to look for the specific aims. So next up is Dr. Tina Morrison from the FDA.
Hi, good afternoon. I'm Tina Morrison. I'm at the Center for Devices and Radiological Health, which today I'm going to tell you about an initiative at FDA, as well as some initiatives on modeling and simulation at CDRH. So when you find this presentation on the wiki, these four pictures will link you to the slide of interest. So I'm going to talk to you about an FDA-wide modeling and simulation working group that we just started, which I'm the chair of. Development tools for both drugs and medical devices, a standard on assessing the credibility of computational models used in medical devices, and a program on applicability analysis. So the modeling and simulation working group, a couple colleagues of mine and I, we started this last year. We actually created a survey that we sent out FDA-wide. We were surprised to get 400 responses from scientists at FDA that said they had expertise in modeling and simulation or were interested in collaborating with people on modeling and simulation. Because of that very broad interest and also the broad expertise from those groups, we decided that it was worth putting together an FDA-wide working group on modeling and simulation. Our main objectives, in particular one personal objective for me, is to really raise awareness around success stories of modeling and simulation used in regulatory decision-making across the agency, specific to advancing regulatory science. But in that same document that we want to work on is to identify challenges and then opportunities. We are trying to create a collaboration environment. And another key thing is to establish credibility principles for modeling and simulation used in regulatory decision-making. What I want to point also out here is the structure of this working group has a leadership circle with members from each of the 7 FDA centers, in addition to 5 interest groups, which are subject-specific disciplines, which will have around 200 members in the interest groups. And then we'll have implementation teams of people who will pull from those groups to develop specific tasks or objectives, such as a communication plan, white paper, and these credibility principles. Another program I want to bring your attention to are FDA's Development Tools Program. So there's a tools program for the Drug Center and also for the Medical Device Center. And there are 3 types of tool programs: clinical outcome assessments, biomarker tests, and non-clinical assessment models, which include in vitro models, in vivo models, and of course in silico models. The key to this program and being successful in this program, if you have a tool that you believe can be useful in the development and evaluation of a drug or a medical device, is to really hone in on the context of use. And this really describes the boundaries of the tool, how the tool can be used, what product area the tool will be used, what stage of development the tool will impact, what is the specific output and role of that tool, and then how can that tool play a role in the regulatory evaluation of that drug or medical device. And the links for those 2 programs are also provided on these slides. another topic that I hear about but I haven't seen much about, and of course tomorrow a lot more of that will become more clear when we talk about credibility. The American Society for Mechanical Engineers has a large committee for verification and validation. This is our charter here. I'm actually the chair of the V&V committee. The committee has 5 subcommittees. The V&V 40 committee is focused on computational modeling for medical devices, and our specific role is to not to tell folks how to do verification and validation of modeling, but to answer a question that is really important, especially to this community, is if I have a specific context of use for my computational model, what level of V&V do I need to achieve in order to demonstrate that my model is actually credible for that specific context of use? So it really talks about 3 key concepts of defining a context of use, assessing model risk, and establishing credibility goals for that model within that context of use. And the end goal is that the rigor of your V&V should be commensurate with your model risk. In other words, how are you using that model to inform some decision? And so there are some materials here. The standard is we hope to publish the standard next year in 2018. In the meantime, there are some materials where you can get a glimpse at what that framework looks like. And finally, I want to leave you with one more program that we're developing at the Center for Devices. Is called Applicability Analysis. It's to complement verification and validation. This is actually outlined in a poster upstairs, number 98. And what I hear in generally speaking about models is I'm a scientist and I develop a model, and when I perform validation, maybe I'm actually someone who works with experimentalists and I can actually get them to perform validation experiments that are really relevant to my model. But sometimes I go to the literature and I use someone else's experiments and I say, hey, I can replicate those experiments, so I've validated my model, so check, I have good agreement, and so now I can take this model, I alter it in some way, and then I use it in another fashion, and then I publish those results. And what we really want to elucidate with applicability analysis is to demonstrate a step-by-step process by which you as a scientist or someone like myself as a regulator can actually look at all the evidence in front of me and determine, is the validation evidence really applicable for how that computational model is going to be used for a specific context of use? And so there are a few examples that are presented in the poster. One specific is looking at electromagnetic modeling of patients who undergo magnetic resonance imaging. So in the short time that I have here, I can't go through the details of that, but you can find more on that poster. So thank you very much.
Thank you, Tina. So next we start our series of NIH lightning rounds. And first up is Dr. Susan Gregurick from the National Institute of General Medical Sciences.
Hi, thanks so much. It's really a delight to be here and to tell you a little bit about computational biology and multiscale modeling at NIH. So I borrowed this figure from a friend of mine to give you a little bit of context where NIH sits. And I imagine that this cartoon is probably not a big surprise to you, but we tend to be more on the more basic side of research and a little bit less on the high-risk side of research and a little bit skewed towards the experienced researcher. But there are programs that are pushing these boundaries in different directions, and I hope that in the future we can show a different type of distribution. So as laid out by Dr. Francis Collins in our FY 2017 priorities to Congress, our mission, of course, remains to extract fundamental knowledge about the nature and behavior of living systems and to apply that knowledge to enhance our health, lengthen our life, and reduce illness and disability. Our priorities for FY17 include enhancing our foundation for basic biomedical research, to deliver on the promise of precision medicine in all of us, as you'll hear tomorrow, to apply big data and to apply technologies to improve human health, and to inspire public trust. So what is NIH spending writ large in computing, informatics, and data science? In 2016, we spent about $730 million of our $31.3 billion budget in a number of computing, informatics and data science areas, including high-end computing, an enormous amount in information management, which would include all of our big data and data sciences, and infrastructure and applications of software and in software design and productivity. So it's not an insignificant amount. All of IMAG does fall under one of these categories, but of course we see a trend that is growing every year as we increase our funding for big data and computing. Some of the initiatives that are supporting computational mathematical sciences include the BISTE initiatives, the BD2K initiatives, which are pivoting to the Commons, IMAG, which you're part of, and our NSF-NIH joint program in mathematical biology. This program follows an NSF solicitation, so there's only one receipt date per year, so please do look at the website for the next receipt date in 2017. I want to tell you about a few FOAs that are in different areas of NIH than NIGMS, and then I'll tell you a little bit about some of the NIGMS activities. Just because there's so much to go on, I can only tell you about a few, but here's one that you may be interested in from NIAID. It's the molecular genetics of drug addiction to encourage applications to identify, validate, and or functionally characterize genetic variations that are associated with addiction. If you load up the PowerPoint slide, you can see a little bit more detail in terms of the receipt dates and when they're needed and some of more details on what they're looking for. I also encourage you to look at the new FOA from NLM to develop novel informatics and data science approaches that can help individuals gather and manage information about their personal health. And when we're thinking about individuals, it's not the researchers that we're targeting, it's the public, it's the patients. So I think this is a new and interesting initiative that you might be interested in looking at. In NIGMS, we have a new program, Maximizing Investigator Research Award, the MIRA Award. This is a very different idea from your typical R01 type of grant. It's one single R35 grant for slightly more money than your typical R01 that will sustain your lab for a 5-year period of time. In this application, you don't write specific aims. You write your vision for what your research lab will accomplish within our NIGMS mission. So there's more flexibility. You don't have to adhere to specific aims. You're doing research that you feel is appropriate and is within our mission. We feel that this will give you a little bit more flexibility and a little bit more time because it's a 5-year time frame. What I like about this program is that it's a stable program to allow you to mentor students and postdoctoral fellows, because once you come up for renewal, it's not a 0 or 1 funding decision. It's a scale funding decision. Your grant goes up or it goes down depending on the nature of your review, and I think that that's exactly what we at NIGMS want to encourage. But if you do take a MIRA award, you cannot subsequently apply for an additional R01 at NIGMS. You're free to apply for other funding at NIH, but not additional R01 funding at NIGMS. So this is it for me, but I have 2 other slides that I have included in my slide packet for you to look at. If you have predoctoral and postdoctoral fellows, we do have some fellowships that may be of interest to them. And thank you very much.
Thank you, Susan. Next up will be Dr. Pankaj Qasba from NHLBI.
So let me tell you, so what I'm going to talk about is actually one of our flagship programs, which is called TopMed, or NHLBI's Transomics for Precision Medicine. So this is a program that started about 2 years ago, and NHLBI has spent a lot of money for the past few years, actually past 3 decades, in many clinical trials, and many legacy studies such as Framingham and Women's Health and Jackson Heart studies. And so we wanted to actually put some of those sample sets that are sitting in the freezers to some good use. And so what this program is really doing is actually taking those sample sets, doing whole genome sequencing on each of them, and then layering using omics, proteomics, RNA, methylation, metabolomics, and trying to see if we are able to pick up some really unique variants in these cohorts. So basically, this program really is to stimulate discovery in high-impact diseases to make sure they are very well phenotyped, and they are very well phenotyped and genotyped. And another most important thing is that this data, once it is sequenced and it's put in the dbGaP, it is accessible to general public, to all your researchers. So basically, is to really build a resource for the scientific community, which hopefully will stimulate you guys to come back and use this data to model stuff and stimulate systems medicine approach. So moving from the genomics to systems biology to systems medicine, and eventually enabling moving forward towards the application of precision medicine. That's where the NIH goals are moving forward. So clearly the goal is really to build resources that has diversity, large sample sizes, and obviously ability to integrate data. We also want to focus on specific diseases, specific patient populations that have not been typically used in the past. We want to, of course, leverage other NIH investments such as Genome Institute's whole genome sequencing and build upon other existing cohorts that are from other institutes and contribute overall to the genomic efforts of NCBI. And so clearly, so this is just some of the basic different sequencing centers. We have an informatics center, data coordination, and archiving all of this samples for now in NCBI, but eventually in dbGaP and NHLBI's data commons, which will integrate and talk to with NIH's data commons in the big scheme of things. So this kind of gives you a perspective of what are the different population sets that we are already in the process of the phenotypic diversity. So we have sickle cell patients, we have stroke, we have asthma, we have COPD, we have hypertension, and of course we also have these multi-phenotype. These are the legacy studies, the Framinghams and the women's health and stuff that I was talking about. Also, the population diversity, as you can see, the typical population diversity in this is predominantly European, and we want to change that so that there is more integration of other populations in this. So why am I talking about all of this to you guys is because we are in the process, we have a different, I'm sorry, I think there's a different slide set here than it was supposed to be. But anyway, so I think there were few other slides in here. I don't see them. Anyway, so I think it picked up a different, it's not, I don't see them here. But anyway, okay, this kind of shows you a work stream of where we are, and I wish, I'm sorry that I'm unable to show the other slides. They don't seem to be, hold on one second, I think, no, this is the wrong slide set. Yeah, it's the same one. I think I had added a different slide set, and that doesn't seem to be showing up here. In any case, what I can do is there were 2 or 3 announcements that are in place. These are the exome mechanisms, where if you have any interesting cohorts that you want to get sequenced or get some omics data on them, I think it said MSM, and it should be at top with MSM. I don't see that here. In any case, so there are some we will put it back on the wiki, the right ones, and so you will have access to those slides. And those slides basically are advertising some of the XO1 mechanisms that we are inviting you guys to look at. And if you have any interesting cohorts that you want to get whole genome sequencing done, or metabolomics or metabolomics or any of the omics components done, will be done free of cost for you, but you have to submit an application to us, and the next available date is in October 2017. There are 2 more receipt dates for that next October. So I would encourage all of you to look at that. It's an amazing resource that I think you as computational biologists and informatics people and engineers can really make use of the data set moving forward. Thank you.
Thank you, Pankaj. So next up is Dr. Michele Ferrante from the National Institute of Mental Health.
Hello, everybody. So this is Michele Ferrante. I'm overseeing currently two programs and the National Institute of Mental Health. One is the Theoretical and Computational Neuroscience program. This program has been around for 25 years. This shows the kind of investment that the National Institute of Mental Health has done in computational modeling. And there is also a new program, computational psychiatry, that is much more recent, was basically created in the last year. The goal of this, so these are two distinct type of programs in the sense that the first one, Theoretical and Computational Neuroscience, is more about basic science. So we try to answer basic science questions with animal models and connecting different units of analysis at different scales. And but the questions are very basic, maybe related to learning and memory, maybe related to normal population. While instead, the second one is trying to bring computational approaches into the field of psychiatry. So the kind of things that we're trying to do is, we are aware that single units of analysis such as genetic information, electrophysiology, behavior can only explain a very small subset of the pathologies that we have in mental health. So we're trying to use something that is called deep phenotyping, that we can basically record the different units of analysis at the same time and try to explain complex behaviors just by integrating this data in an intelligent way. So there are several priorities in this program, and one is trying to understand if we have a multi-scale model that is also multimodal in the kind of things that in the kind of inputs that it takes in, what is the brain malfunction, or what is the failure mode for specific mental health pathologies such as schizophrenia, depression, or any of our priorities. So one of the we are integrating at many levels. So for instance, if we are coming in with something that is a deep learning model, we would like for this model to also provide explanation on all the classification and predictions. And there is a big focus on integrating theory and data-driven models at the same time. Same is true for bottom-up and top-down models. So what we are trying to do is models that not only can capture the dynamics and can make prediction, but it can also basically mechanistically explain the biology that is underneath. Because the idea for us is that this model needs to be eventually translated in the clinic. So my background is in biophysically realistic models. So these are models of neurons that can go all the way down from the genetic expressions and go up to the molecular proteins, neural morphology, and signal integration, systems level neuroscience, all the way up to behavior. So I wanted to talk to you about 4 funding initiatives that are going on right now. One is trans-agency between NSF and NIH, and international between France, Germany, Israel, and obviously the US. So this is called the Collaborative Research in Computational Neuroscience, CRCNS program. If you're interested in in this kind of effort, you should contact one of us here in NIH. What you should know is that it's a tight program that stimulates collaboration with the experimentalists and theoreticians, and it's capped at $250,000 per year and for 3 years of funding. We have also a temporal dynamics neurophysiology pattern. This is like an R1 project that is trying to understand how rhythms in the brain affect brain function and behavior. And we're very excited about the third initiative. This is like a collaborative UO1 project where we're actually looking at convergent approaches that start from genetics and can use like several units of analysis to explain the same type of behavior. So we're trying to basically converge in the same, with different units in the same problem. And finally, if you know anybody that has a grant at the National Institute of Mental Health and you are a computational modeler and that person is an experimentalist, I would encourage you to contact that person and to ask to start a collaboration on one of our latest efforts, which is basically the release of computational supplements for a regular NIMH grant. And with this, I actually don't have anything else to say. Thank you, Ed. Thanks.
Thank you, Michele. Michele and I actually work together both in IMAG and on the BRAIN Initiative, so you can ask him about that when you visit him. Last but not least is Barbara Sorkin from the Office of the NIH Director. And here's your talk.
It's a pleasure to be able to tell you this afternoon about some opportunities for modeling in conjunction with the NIH Centers for Advancing Research on Botanicals and Other Natural Products, the NIH Carbon program, which I co-direct. The program currently consists of 5 centers for a small office. The first 3 of them are focused on comprehensive characterization of botanicals and the comprehensive chemical characterization and their effects on human health. You can see that they're looking at 3 quite different areas of health outcomes. The last 2 bullets on this slide show 2 different flavor of the center program that's focused on the development of new approaches and methods for analyzing natural products and their health effects. I talked about comprehensive chemical characterization because we're interested in, missing a slide here, Grace. Can I escape out? Okay, well, we'll use this one. There was one in between, it's fine. I wanted to give you a flavor of the chemical complexity of it's not there. It didn't let it. It's just not there. Okay, doesn't matter. Sorry. A lot of different compounds in many of these botanical preparations, including structurally unassigned compounds. There's a tradition among herbalists in asserting that using the complex mixtures has advantages. Those of you who work with infectious disease know that if you have multiple mechanisms of action, multiple actives, that can certainly be advantageous. This is an example comparing, let's see if I can get the mouse to work without, there we go. Comparing artemisinin, the WHO frontline drug for malaria, with a whole plant preparation containing an equivalent amount of artemisinin, and you can see that the whole plant preparation is considerably more effective in suppressing malaria parasitemia. Obviously, there are additional compounds in the whole plant preparation that contribute to anti-malarial activity. You can see where that would be advantageous in slowing, and in fact, this group has reported more recently, it does seem to be advantageous in slowing the development of resistance. So what do you do when you're adding on to the biological complexity that all of you are familiar with modeling? a complex product with potentially unknown actives in it. That's where our methodology centers are beginning to do some modeling in this area. This is an example of work from the last one on that slide. They are integrating data from mass spec of these complex mixtures, different fractions, with data on standard RNA gene expression omics, with cell phenotype, high throughput cell phenotype data. And they have shown that with marine microorganisms, they can successfully use this to point to both chemical component interactions and novel signaling networks and pathways that you wouldn't necessarily get using the omics approach. This, however, is really, and then there's the additional, yeah, it's in there, of course, dynamic complexity that you get when you're metabolizing orally consumed complex mixtures. There are a lot of reports recently, growing number, that there is a role for the gut microbiome in generating active metabolites of botanical products. So there are opportunities for collaboration with all of these centers, in particular the methodology centers. They're very eager to collaborate with people who are interested in working to help them develop their methods, as well as beta test them. In addition, we have funding that I think would be ideal to supplement certain existing NIH awards. Would be ideal, I think, to work with investigators currently doing research to collect the kind of data that would be needed to begin to develop good models for how these products are working on human health. So I think that's all the information you need to find, and thank you very much.